The overall goal of this procedure is to monitor ethanol levels in brain dialysate during an ethanol self-administration session in order to compare this measure to synchronous self-administration behaviors. This is accomplished by first surgically implanting a microdialysis guide cannula and tether bolt, and then training rats to operantly self-administer ethanol. The second step is to habituate the rats to the head stage tether so that it will not impede ethanol self-administration during microdialysis. Then, microdialysis is performed during an ethanol self-administration session, during which a portion of dialysate is prepared for ethanol analysis. Next, the dialysate samples are analyzed for ethanol concentration using a gas chromatograph with flame ionization detection. Ultimately, the results obtained show the time course of ethanol reaching the brain region of interest during an ethanol self-administration session.
The main advantage to this technique over existing methods like tail or saphenous vein puncture is that this procedure is minimally disruptive at the time of sample collection and allows for drug measurement over time in the brain region of interest while the animal is performing a behavioral task.
Generally, individuals new to this method will struggle because it requires a number of specialized skills, many of which can only be acquired through repeated practice or experience. Also, demonstrating the procedure will be Geoffrey Dilly, an undergraduate research assistant in our laboratory.
All animal procedures shown are approved by the University of Texas Institutional Animal Care and Use Committee and follow all Guide for the Care and Use of Laboratory Animals regulations. The University of Texas at Austin is an ALAC-accredited facility.